Hello anatomy students. Today I wanted to give you an overview of cellular division. We're talking about the difference between mitosis and meiosis because we are talking a little bit about how sperm develop and how ova develop, and it's important for you to know the difference between these two types of cellular division and how the genetic material is divided. So here are our objectives. Identify which type of cell division is required for somatic cells and for gametes. Describe briefly the events of mitosis and meiosis, and explain the purpose of each type of cell division. All right, so two kinds of cell division, mitosis and meiosis. Let's just look at a brief comparison here. Mitosis occurs in body cells, whereas meiosis occurs in gametes, meaning the cells that form the eggs or the oocytes in females, and then the sperm in males. Mitosis is going to produce only two daughter cells. There's only one cell division that occurs, whereas meiosis produces four daughter cells as a result of two cellular divisions. Mitosis is going to produce two daughter cells that are exact genetic copies of the parent cells, whereas in meiosis you have some genetic variation happening because of something called crossing over that occurs before the first cellular division. Daughter cells after mitosis are diploid in number. They have 46 chromosomes, whereas daughter cells as a result of meiosis, these are the actual gametes, the actual oocytes and the sperm. They are haploid. They have 23 chromosomes. And this is really important because when you have fertilization occurring, when a sperm joins with an egg, then you just add their chromosomes together so that the resulting embryo has a total of 46 chromosomes, because 23 plus 23 is 46. And that's what I was just talking about. A gamete plus a gamete, a sperm plus an oocyte produces a diploid cell, 46 chromosomes, 23 plus 23. All right, so let's talk a little bit first about mitosis. I think that part of the confusion about cellular division is the use of the word chromosome, because as you can see on the slide, this is a chromosome. It's made up of a single chromatid. So that is a chromosome. But also on this slide, on this picture, we have chromosomes that look a little bit different. This right here is a duplicated chromosome. So it's still a chromosome, but it has twice the amount of genetic material because it has duplicated itself. So we have chromosomes that are made up of one chromatid, and then here we have a duplicated chromosome that is made up of two chromatids. These two chromosomes that we have here, you can see that they're the same length. One is black, one is gray. These are called homologous chromosomes. One is coming from the father and one is coming from the mother. So that's one chromosome pair, one from mom, one from dad. These are called homologous chromosomes. And then during mitosis, our chromatids duplicate to form duplicated chromosomes. Notice that the sister chromatids are attached in the center by a centromere. And then you have cellular division, where one chromatid from each duplicated chromosome is moving into a separate cell. And so what's happening with the genetic material is shown here at the bottom. Again, you have chromosomes that are made up of single chromatids that are found in the nucleus of daughter cell 1, and chromosomes here are found in the daughter cell too. And here you can see that the cell has duplicated here. So we've got 2 daughter cells that have exactly the same genetic material, the same chromosomes as what was in our parent. So we have 2 daughter cells that are exact copies of the parent. Now, in meiosis, which again is happening in gametes, there are a couple of things that happen before the first cellular division. So once again, we are starting off with our homologous chromosomes here. And remember that this is a chromosome. It's made up of 1 chromatid. We have chromosome duplications, and we get duplicated chromosomes that are made up of sister chromatids or 2 chromatids, and they are connected to each other by a centromere that's in the center. They're going to form tetrads. These duplicated chromosomes form tetrads, which means that the duplicated chromosomes of the homologous chromosomes, 1 from each parent, are going to come together. Their centromeres are going to join, and they form this tetrad, which is 4 chromatids that are connected to each other in the middle by the centromere. And then a phenomenon called crossing over happens, where you get 1 piece from one of the sister chromatids switching places with a piece of an adjacent sister chromatid. And so that's what I'm showing you here in this diagram. So you can see that a portion of the black chromatid has moved over to the gray chromatid, and a portion of the gray has moved over to the black. And then it's the same on all of these. So here you have a portion of the lighter chromatid jumping over to the darker chromatid, and vice versa, and it's the same over here. And so now if you look at all of these, none of the chromatids are the same anymore. You have a whole black one, you have gray and black, black and gray, and then all gray, and then you can do this, you can look at the other ones and see the same thing. So now none of the chromatids are the same as they were when they started out. All right, so this is all happening before the very first cellular division. So let's see what happens when the cells divide. All right, so here we have our tetrads with all unique chromatids. And now we get our first cellular division occurring after the homologous chromosome separates. So now the tetrad is going to separate so that when we're done, we still have duplicated chromosomes, but again, they're all different because of the crossing over has happened. And this is what happens during our very first cellular division here. So when we get our first cellular division, our sister chromatids are staying together. We still have our duplicated chromosomes, and we get 2 daughter cells. Each daughter cell has 23 chromosomes. Now, granted, they're duplicated chromosomes, but they're still considered chromosomes. So we've gone from 46 chromosomes down to 23 chromosomes. So the very first cellular division in meiosis is considered reductional. We've reduced the number of chromosomes in the first cellular division. We've changed from 2n, which is 46, to n, which is 23 chromosomes. We're not done. Meiosis 2 refers to the second cellular division and the formation of gametes. So in meiosis 2, we're starting off with our duplicated chromosomes. Each chromatid is unique, but they're still joined in the center by the chromatids. So the sister chromatids separate, and we get 1 chromatid for 1 chromatid is going to go into each cell. So we get 1, 2, 3, 4 daughter cells. So this is cell 1, this is cell 2. Cell 1 divides and cell 2 divides in meiosis 2, and what we're left with are 4 daughter cells which have produced the gametes. And if you look, we still have 23 chromosomes, so meiosis 2 is considered equational division. We have not reduced the number of chromosomes here. And our gametes are haploid, meaning that they have N number of chromosomes, or 23, and they are genetically unique. If we look back at our bigger picture here of the chromatids that we have, if you look, no two chromatids are the same, and no daughter cell has the same genetic material as another daughter cell. This genetic uniqueness is happening because of the crossing over that has happened when we had the tetrads. And then because we have 2 cellular divisions, we have unique daughter cells. Each daughter cell has a unique genetic composition. So when a haploid gamete, let's say from the father, from a sperm, joins or fertilizes the oocyte from the mother or the female, we've got N number of chromosomes here, or 23, and N number of chromosomes here, another 23, and 23 plus 23 is 46. So when they combine during fertilization, you get a diploid cell, and that is what the zygote is. And so from here, during embryonic development, from here you get mitosis occurring, and that's how the body cells are produced in the growing embryo and throughout life. They undergo mitosis. So remember that meiosis is going to yield different results in males and females. So in the male cell, you get 4 daughter cells, again, genetically different, but they're all the same size. Whereas in a female, you still get 4 daughter cells, but you get 1 viable oocyte, 1 cell that is able to be fertilized, and you get 3, they're called polar bodies, and you get 3 cells that usually die off. They are not viable. They're not going to have the correct amount of cytoplasm that they need in order to survive. All right, let's see what you remember here. Which type of cellular division produces gametes, mitosis or meiosis? Your answer, meiosis. Which type of cell division results in exact copies of the parent cell? Your answer, mitosis. And 3. Which process results in 4 daughter cells that are genetically unique? Your answer, meiosis. All right, this is what we'll tackle during class. Thank you for watching, and I hope you have a great day.